Good morning, my dear students. Today, the topic is going to be the biofertilizers. In the last two classes, we were discussing about the role of biology in human welfare. Even though human himself is a component of biology which includes plants and animals, we the human maximum utilize the other organisms for our benefit. Our dependency on plants and animals is to a very maximum extent. The other plants and animals also depend on the human being for their sustenance. But still, our dependence on the plant and animal is more, and if I say this, I think it will not be an exaggeration. All the plants and animals in this world can live without human, but no human can live without the plants and animals. To that extent, we are depending on the plants as well as other animals. So, what is the role of biology, that is the plants and animals, for the human welfare is what we are studying or learning in these last 2 days. we are making a lot of changes in the crop production and also in the animal production, and it has resulted in the production of more crops, better crops, economically viable crops, commercially viable crops. So this is what we have done in the last 100 years to improve the quality of the crops. How we have done this is what I was telling you in the last two classes. And in today's class, we are going to study a little bit about fertilizers. Now, the fertilizers are of two types. One is a chemical fertilizer and another one is a biofertilizer. Now, to start with, man was using only a biofertilizer. He was using only a he didn't know anything about the chemical fertilizer, because when a plant it has depleted all the minerals from the soil, they understood that the soil has become stale and it is not able to support the crop production anymore. So they had their own technique of preparing the fertilizers. These fertilizers they made from the other plants. There are many plants which act as a fertilizers. They cut down the branches, they bury them under the soil, add salt and so many substances also to promote the process of decaying and decomposing, and finally, say about in a period of 2 months or 3 months, the fertilizer will be ready to be given to the other plants or to be distributed or to be fed to the next crops. So this is how we prepared our own fertilizers for our plants. But at a particular stage, what happened? People thought that these fertilizers that we make from the animal extracts and plant remains will not be sufficiently supporting the life of the crop plants. That's what we thought. And so, if we are adding the chemical manures directly, then it will be able to give a better food production. But what happened over a period of long 50 years, we have understood that instead of giving or making the soil fertile, the chemical fertilizers have damaged the soil equally. because when you are adding nitrogen, phosphorus, potassium, what we popularly call as NPK, it will be an instantaneous source of energy for the plants, no doubt about it. It will instantaneously supply the energy, but in a long run, the soil is impregnated with these chemicals and it becomes unsuitable for supporting the life on it. So once again we had to come back to what is called as biofertilizers. So what is a biofertilizer? A fertilizer of biological origin. Biological origin means what? It could be from a plant, animal, microorganism, and even algae, fungi, bryophytes, pteridophytes, etc. So anything of a plant origin or animal origin is called as a biofertilizer. So a fertilizer derived from the other biological organisms, that is what is called as a biofertilizer. So what is the role of a biofertilizer? First we will go to the definition. Biofertilizer denotes all the nutrient inputs of biological origin, particularly nitrogen compounds for plant growth and yield. So biofertilizer, it denotes all the nutrient inputs, whatever nutrient we are giving to the soil, it refers to that, and its origin is a biological origin. Particularly it will be supplying the nitrogen compounds, then it becomes a biofertilizer. It includes, what are the different components of the biofertilizer? It includes bacteria, fungi, and cyanobacteria mostly. Okay, it includes bacterial origin, fungal origin, and cyanobacteria. Producing nitrogen compounds, they are known to fix atmospheric nitrogen into metabolically usable compounds. Nitrogen fixing bacteria include Azotobacter. This Azotobacter is a free living bacteria and it includes Rhizobium also. Rhizobium is the best example for a symbiotic bacteria. So what is a free living bacteria and a symbiotic bacteria? A free living bacteria live freely in the soil and a Rhizobium, it lives in association with the leguminous plants. It is one of the best examples for the symbiotic association. So it lives in the root nodules of the leguminous plants. So both of them are helping in fixing the nitrogen. So one is a free living, another one is in a symbiotic association. Okay. So biofertilizer denotes to the nutrient outputs mostly in the form of the nitrogen compounds into the soil, and this is being derived from the bacteria, fungi, and cyanobacteria. Okay. So I have just given you a very simple classification of the biofertilizers which will help you to understand what a biofertilizer is. So I divide the biofertilizer as a free living and symbiotic. In the last slide I was telling you what's the difference between the free living and symbiotic. Free living, they live mostly in the soil, and the symbiotic means it is in association with the other plants. Okay, fine. So what are the free living bacteria? They belong to two categories once again. They are blue-green algae, Anabina, nostoc are the examples. They are the blue-green algae and they are Anabina or nostoc are the examples. Then bacteria is another free living example. Now I have given two examples here, Azotobacter and Azospirillum. Here I have given two examples, Anabina and nostoc. So for free living, Cyanobacterium I have given 2 examples, and for bacteria I have given 2 examples. Okay, that's fine. Now I am going to the symbiotic association. Now symbiotic bacteria they live in the leguminous plants. For that the example is Rhizobium. They live in the non-leguminous plants also. Example Frankia. Okay, this is for a Casuarina. Now in addition to this free-living bacteria or free-living cyanobacteria and symbiotic bacteria, there is a mycorrhizal association also. This mycorrhizal association you can divide into ectotrophic and endotrophic. All these things we are going to see in a very detailed way. I will go to the explanatory part of that after some time. Ectotrophic mycorrhizal association is the presence of the fungi on the outer surface of the plant. Endotrophic means the presence of fungi inside the plant, inside the tissue. That is called endotrophic. So, okay. So, in this slide, what I am trying to tell you, biofertilizers they are derived from the microorganisms, and this I have divided into 2, namely free-living microorganism and symbiotic. Free-living, once again, I have divided into cyanobacteria and bacteria. Cyanobacteria, the example is Anabina and Nostoc. Bacteria, the examples are Lactobacter and Azospirillum. And symbiotic association could be by a bacteria or fungi. Bacteria could be once again leguminous, non-leguminous. Leguminous example Rhizobium, non-leguminous example Frankia. Then mycorrhizal association, the association of the fungi with the other higher organism, this could be exotrophic and endotrophic. Fine. So this is what you are able to see. This is this is present in the root nodules. See, this happens in the cycas. What they have given, what we popularly call as a coraloid root. The coraloid root, a beautiful structure that we are able to see. The roots become divided into two at every stage, and then it looks like if this is the root, it will divide into 2 like this, and then it will divide into 2 like this. At every stage it will be going on dividing into 2 and 2 like this. As a result of that, it will look like a coral. Okay, so that is why it is called as a coraloid root, that you are able to see beautifully here. See, this includes nodules. The bacteria in the night, they are able to fix the nitrogen, and they are able to convert them into ammonia, nitrates, etc. They are in the root nodules of the leguminous plants. This is the root of a leguminous plant and these are the lateral roots, and in these lateral roots you are able to see the nodules. And these nodules contain the bacteria. Okay, and they help in the nitrogen fixation work. They convert the nitrogen into nitrites and nitrates, and then also into ammonia. Now you see certain cyanobacterium algae. The best examples are Nostoc, Anabina, and then some more, what is called as Oscillatoria. Very good example is that Oscillatoria is also a cyanobacterium algae. All these they possess what are called as echinates and then heterocysts. These echinates and heterocysts, they are responsible. Echinate is normally a reproducing structure. Only heterocyst is doing the function of nitrogen fixation. Okay, so these cyanobacterium algae also help in the nitrogen fixation. See, this is a beautiful diagram on the ectotrophic and endotrophic type of association I was telling you. These are fungi which are living in the soil. This is the root hair, and surrounding the root hair you will be getting the soil. These are the soil particles, and fungi grow in the soil. It's a very good example of a soil fungi. Now this soil fungi, the fungi grows in the soil and then it enters into the plant, and then it goes into the cell, and in that cell it produces what is called as arbuscules. So this is the epidermis of the root, and this is the cortex portion of the root. It enters through the epidermis and then goes into the cortex portion, and then the fungus starts reproducing. Now it develops from a chlamydospore. It develops from a chlamydospore. This is the external hyphae. From this chlamydospore, the fungi is growing, and then it is entering into the plant through the epidermis, travels inside, invades the cortex portion, and then it starts colonizing there, and then this becomes very much beneficial for the higher plant. These are all helping in the nitrogen fixation work. So here the fungus is having a lodging arrangement. It goes into the higher plant and then it is living there, but at the same time the plants are benefited by that. This higher plant, it is able to give protection to the fungus, and at the same time it is benefited by that. And this fungus, it goes and then it finds a place for it to live. So we call it a sort of a very good association. We divide the association between the plant and other organisms as a positive and negative, and these are all coming under what is called as a positive associations. So positive association means it is good for the fungus and it is good for the plant also. Both are equally benefited. Okay, so this type of association we are able to see in the some of the plants related to fungi. See the root nodules of a particular leguminous plant. See the root system. I used to tell, see, in what we are seeing only as a small plant. This is what a man sees about a plant, and he thinks that the plant is very small, but the hidden treasure is inside. That is what is called as a root system. See how big the root system is. For this small plant, it has got such a big root system. It is the root system which is supporting the plant, an unseen treasure for a plant. Okay, so this root system, it is extensively branched, so extensively branched, and then you have got the root nodules present there. So this photography shows very beautifully how a small plant will have an extensively branched root system is. Now at this situation, I will tell you one very important thing to tell you the extensivity of a root system. Now this is a maize plant. This is a maize plant. Okay, and then it has come into the flowering. It has produced the crops, etc. Now the height of the plant is 5 feet. A human height. A human height 5 feet. And you see, a maize plant is what is called as a monocotyledonous plant, and then it is producing a fibrous root system. You have got a fibrous root system coming from the base of the maize stem. Okay. Now what I am doing, I am removing these fibrous roots. I am removing this and then attach one after another and then put it in a line. I put it in a line like this. All the roots I have removed, and then I try to join one after another, and then I make it as a long line. Now can anybody tell me what will be the approximate length of this root that I constructed by removing the fibrous root? Now we understand what I have told you. The plant is 5 feet in its height. I have removed the roots, and then I have joined the roots by placing it one by the end of another, and to make it as a look like a long rope, something like that. Now my question is, what will be the length of this root that I have made? when I put this question to a class, when I am teaching in a live class, some student told it will be 5 feet, sir, because the plant is also 5 feet. But some students told 5 meters, sir. 5 meters, good. Some students told 500 meters, sir. 500 meters. Different answers they gave. But you know what the real answer is? It is 5 kilometers. 5 kilometers. That is a 5,000 meters. A plant which is having only 5 feet in its visible area over the ground, it will be having 5,000 meters of the root system. It is so extensively branched, and in some of these plants it will be having the root nodules, etc. So it's a very beautiful information that each and every one of you should know before we proceed to the next slide. Fine. So the microorganisms which are present in the soil, they increase the crop yield by 20%. Pseudomonas striata, which is a bacterium, is used as seed inoculations as biofertilizer coats for seeds of cereals. Various leguminous plants, example, Crotalaria gentiana, Glycine max, Indigofera tinctoria, Indigofera linifolia, Sesbania prostrata, Acacia nilotica, Lysina glauca, and Mucuna. So, what are the plants which are used for biofertilizer? Shall I pronounce once again? Crotalaria gentiana, Glycine max, Indigofera linifolia, Sesbania rostrata, Acacia nilotica, it is not Lysinia, it is Lenzena glauca and Mucuna. So 7 plants are there, in whose roots the nitrogen bacteria fix atmospheric nitrogen, and they are used as green manures. They accumulate more than 80 kilos of nitrogen per hectare. See? Beautiful. They accumulate more than 80 kilograms of nitrogen per hectare. Per hectare contains 80 kilograms of nitrogen fixed because of these plants. Okay. So where is the leguminous plants? Crotalaria, once again I repeat, if you find it very difficult to pronounce, because you see certain the name of the plants and animals, for the beginners it looks very complicated. It looks like a jargons. They are not able to pronounce the words of the plants and animals properly. We teachers, we also sometimes find it difficult, and you students find it very difficult to begin with to pronounce the name. So whenever a teacher is pronouncing the name of the plants and animals, you please repeat with them. And so that you will be getting the correct pronunciation, and then you will be able to pronounce it once again even in the absence of a teacher. So for your benefit I am repeating once again. So the name of the leguminous plants which are used are Crotalaria gentiana, Glycine max, Indigofera linifolia, Sesbania rostrata, Acacia nilotica, Lenzena glauca, and Mucuna. So these are the plants which are used. Now we take the presentation on some of the algae which are used as a biofertilizer. So the best example is Anabina azole. So why, first I tell you why it is called Anabina azole. There is a plant called azolla. It is a pteridophyte. Now this pteridophyte, in the leaves of this pteridophyte, these cyanophysian algae, Cyanophage and bacteria, or you can call it is coming under algae also. So they live in the leaves of the pteridophyte. So that is why the name of the cyanophage in alga is Anabina, and where it is residing in the leaf of the azolla, so it is called azole. So whenever the name of the nomenclature is given to a plant, whenever a name is given to the plant, there are certain rules governing the nomenclature. See, for example, if a bacterium is a disease causing bacterium, then the generic name, the genus name will be the name of the bacterium, and the species name will be the type of the disease it is caused. See, will be having tuberculosis. It will be causing tuberculosis. So it is called tuberculosis. Like that, the species name will be based on the name of the disease. Pyricularia oryzae. Pyricularia is the name of the fungus. It is causing a disease on the oryzae. So it's called a Pyricularia oryzae. So it will be easy for us to remember the things. So Pyricularia is causing a disease on the oryza. So that particular fungus is called as a Pyricularia oryzae. Okay, like that here this plant Anabina is living in the azole. So it has been given the species name azole. So Anabina azole is an endophytic cyanobacterium occurring in the leaves of Azolla pinnata out of 6 species of Azolla. Because of this, it is used as a biofertilizer in Indian paddy fields. It adds 30 kilograms of nitrogen per hectare, where yield is equivalent to that of urea and ammonia phosphate. See, this point I was telling you even in the beginning. At a particular stage in 1950s, forgetting about our biofertilizers which we were traditionally using, we switched over to the urea. And ammonium biofertilizers. This we started using in 1950s. It's going to be nearly 70 years or a little less than 70 years. And over these 70 years, we have learnt a lot of lessons. We have understood how bad this urea becomes, how the soil will be made important because of the constant use of the urea and ammonium phosphates. So now we have started abandoning these urea and ammonium phosphate. So you cannot underestimate the role of algae in the agriculture, because I am giving you a statistical information. It adds 30 kg of nitrogen per hectare, which is more or less equivalent to that of the urea and ammonia. So don't think that urea and ammonia will alone be able to improve the crops. These are all chemical fertilizers. What the chemical fertilizers are doing, equal potentiality can be given by the biofertilizers, but at the same time the negative aspect of the chemical fertilizer will be avoided here. Okay. Then fungi which are used as biofertilizers. Mycorrhiza is a root inhabiting fungus occurring around ectomycorrhizal association or inside endomycorrhizal association, the roots of many plants. This is of 2 types. Ectomycorrhizal association, it occurs outside the surface of the plant roots. Example, Basidiomycetes fungi. Now, in fungi, when you go for a classification, if it's a regular fungi class, I will tell you. It is divided to Myxomycetes, Ascomycetes, Phycomycetes, Basidiomycetes, Deuteriomycetes. It is divided into 5 major classes: Myxomycetes, Phycomycetes, Ascomycetes, Basidiomycetes and Deuteriomycetes. So this ectotrophic mycorrhiza is a very good example of a Basidiomycetes fungi. Then endotrophic mycorrhiza, it is found inside the roots of worm. That is what is called as it could be intracellular or intracellular. These are popularly called as a worm. Worm means vesicular orbiculaar mycorrhiza. Vesicular or orbiculaar mycorrhiza. Now I have to explain to me what an intracellular or intracellular, that I will do. So endotrophic mycorrhizal association is once again divided into intracellular and intracellular. Okay, fine. What is these? Let's imagine these are the cells of the. Sorry, this is cell. This is wrong. These are the cells of the plant on which the fungus is living. These are the cells of the fungus which it is living. Okay. Now, if the fungus is living within this, then it is called as intracellular fungus. Intra means within. Okay. Now, if the fungus is living in between the cells here, these hyphae and all will be here, and then it will be living in between the cells. Okay. It won't go inside the cell. If a fungus is living like this, it is called intercellular. Intercellular, exactly. Inter means between, and this is intracellular. So the endotrophic association, endomycorrhizal association could be intracellular or intracellular. Okay. Ectotrophic association will always be on the surface. It will be only on the surface. Then the benefits of biofertilizers. They are easy to produce and are available locally at low cost to the farmers. This is very important. When you compare the cost of a biofertilizer to the chemical fertilizer, now we are able to produce the biofertilizer at a lower cost. So it is available to the farmers locally. That's another important thing. A farmer himself can prepare a biofertilizer. A farmer can never be able to prepare a chemical manure, whereas a farmer can prepare, he can make his own biofertilizer, provided if he is getting some plants and animals which will be used for making the manure. Okay. They increase the soil fertility without any pollution. That's more important, very, very important. The biggest problem nowadays in our country is a pollution. So a biofertilizer will be causing no pollution, but whereas a chemical fertilizer will be damaging the soil. If you are going to use it continuously for about 20 years, 20 years is more than sufficient to spoil the soil. Okay. They increase the soil fertility without any pollution to soil and atmosphere. Azolla, with its cyanophycean algae, anabino in their leaves, amends the soil with organic matter. They secrete plant growth regulators, such as IAA, indole acetic acid, indole butyric acid IBA, naphthalene acetic acid NAA, and amino acids, protein and vitamins to soil. So they secrete the growth regulators, growth promoters, and then they are able to control, that is, they are able to produce IAA, IBA, NAA, then ammonia, amino acids, protein and vitamins to the soil. It is able to enrich. They are biodegradable. That's very important, very, very important. So biomanures are biodegradable. When you add it to the soil, chemical manures, it will be there for a quite long time. These chemicals will be there. and that will be making the soil sterile after a long time. Whereas these biomanures, biofertilizers, they are biodegradable. They are biodegradable and cause no hazardous residues to the soil, or they don't leave any hazardous residues in the soil. Now, cyanobacteria is a neutralizer, is neutralizing both acidic and alkaline soil. Blue-green algae convert the untenable fallow land to cultivable land. That is what is called soil reclamation, a beautiful concept in botany. Soil reclamation. What is a soil reclamation? When the soil becomes suitable for cultivation due to the damage that has been done to the soil because of the chemical manures, you reclaim the soil. That is, you bring it once again to its virgin form. You bring it once again to the cultivable form. The land should become once again cultivable. So, a fallow land should be made as a cultivable land, and this process is called as a soil reclamation. So, an untenable fallow land, when you are converting into a cultivable land, it is called as a soil reclamation, and this soil reclamation is being done by cyanobacteria. So, cyanobacteria is a neutralizer, because when you add chemical fertilizers, chemical manures, it becomes either acidic or alkaline. When you grow cyanobacterium in that particular soil, it is able to neutralize both the acidic substance as well as alkaline. It's a very beautiful thing that to be discussed in chemistry. When you add a chemical to another chemical, either it will be able to neutralize acidic substances or alkaline. It cannot do the both. But you see, the biological products, one single substance is able to neutralize it is both acidic as well as alkaline, which are present in the soil. That is the benefits of using the biofertilizers. Nitrogen fixing bacteria, whether it is aerobic or anaerobic, add about 50 to 150 kg of nitrogen per hectare. The examples are Acetobacter and Azospirillum. They are free living bacteria. They live in the soil. They may be aerobic, that is they live in the presence of oxygen, or they could be anaerobic, they are able to live in the absence of oxygen. They add about 50 to 150 kg of nitrogen to the soil per hectare, and secrete antibiotics which act as a biopesticide. See, you need not to put any antibiotics into the plant. They absorb these antibiotics through their root system, and then they act as a biopesticide also. So, a biofertilizer is also acting as a biopesticide. See the benefits of a biofertilizer. The biofertilizer is not merely a biofertilizer. It also becomes a biopesticide for the plant because it is able to secrete certain antibiotics. So, it is improving the quality of the soil. It is also able to prevent some of the diseases which are coming to the plant by the antibiotics which are secreted by these nitrogen fixing bacteria, which could be aerobic or anaerobic. And the examples I have given you, they are the Acetobacter and Azospirillum. Then heterotrophic mycorrhiza increases the surface area of the roots of the host plants. They are responsible for more absorption of nutrients, but there is a particularly phosphate by roots. It increases yield up to 45%. You see, increase up to 45%. You understand what do you mean by this term 45%? If you are getting, let's imagine 100 bags of rye, paddy or wheat, whatever it is, doesn't matter. 100 bags of a particular product, 100 bags of black gram, 100 bags of green gram, or 100 bags of paddy, anything. So if you are going to use this biopesticide, this 100 will become 145. 100 bags, instead of 100 bags, will be getting 145 bags of paddy, wheat or black gram, whatever it is. That's what we mean by 45% of increase. So it increases the yield up to 45% well over chemical fertilizers in the soil. It increases the nitrogen as long as they remain in the soil. Now, plant breeding for resistance to insect pest. Now, insect resistance in crops may be due to the morphological and biochemical. There are two, I am dividing into two. So, this insect resistance is offered by the plant to the insects. Some of them are possessing the hairy leaves in several plants. They have got hairy leaves. So, when the leaves are becoming very hairy means if you just count the number of hairs per millimeter square. See, I am just writing it for millimeter square. Each and every millimeter square will contain approximately about 20 to 30 hairs. So tufted they are, so clustered they are. It will be just like a blanket over the leaf. That type of leaves are also seen. It will be completely, totally covering the leaves. So any insect cannot come and then automatically sit over the leaves. You could have seen some animals also. They have got a very thick skin, they have got a hairy skin, because they want to prevent themselves from the other pests, most like mosquitoes and all. See, we the human being, we take some precautionary measures to prevent ourselves from mosquitoes. Whereas animals, what they will do? Only nature has to protect them. So, the nature has provided them with a very thick skin, hairy skin, so that the pest will not be able to have a contact directly over the skin. Okay. So, hairy leaves in several plants. Resistance to jazites in cotton. So, that is the name of an insect. So, it is able to resist these insects because it is able to produce some sort of a resistance. Then, cereal leaf beetle in wheat. It is able to prevent these beetles, cereal leaves. Solid stem in wheat. See, the stem becomes very much solid. They are not very soft. So, when the stem becomes very solid, the soft fly cannot bore into the stem and then it can't do any damage to this wheat plant. So, this wheat plant is having a solid stem to prevent the soft fly entering into that. So, these are all some of the resistance, the mechanism of resistance offered by the plant to prevent the insects entering it. Then, smooth-leaved nectar, smooth-leaved and nectarless cotton varieties. When you are having nectar in the flower, the insects will automatically come. Okay. So, the nectar will attract the insects. So, the flower will not be having any nectar. So, that an insect, the opportunity of a leaf of an insect coming to the plant is made very less. Okay. So, this is a so, this resistance to the insect is offered by biochemical and physiological characters also. First we discussed the morphological. Previous slide was on the morphological adaptations. This slide is on the biochemical and physiological characters. Now what are the physiological characters? They have got a high aspartic acid, then low nitrogen and low sugar content in the maize plant for maize stem borers. Maize stem borer is an insect. This maize stem borer will be boring the maize plant and it will be damaging the plant. So if this maize plant is having a low nitrogen and a low sugar content, then it may not be able to do. The maize stem borer will not be able to bore the maize stem. So these are all some of what are known as the biochemical and physiological adaptations seen by the plant to resist the insects. Breeding methods involve the same steps as those for any agronomic trait. See, when you want to produce a better quality crop, when you want to produce a disease resistant crop, or we are breeding the plant with so many aims in our mind: disease resistant, a pest resistance, insect resistant, salinity resistance, like that. For so many characters we are doing the plant breeding experiment. So the plant breeding procedure is the same here also. There is no difference. So the method involved is the same. Sources for the resistant genes, disease resistant genes, insect resistant genes, pest resistant genes, these are all taken from the cultivated varieties or from germplasm or collection of cultivars or from the wild varieties. I have already told in one of my lectures that wild varieties will have disease resistant qualities. So they are taken from these genes are removed, or they are taken, the wild varieties are taken and then they are crossed with the modern varieties, so that they will also derive these disease resistant characters from the wild relatives or wild varieties. Below are some of the released crop varieties by the hybridization selections. So the crops selected is brassica, mustard, then flat bean, and then the okra. There is a bindi. Bindi means lady's finger. We call, lady's finger. Vendekkai. Okra. So the variety used for breeding is Pusa, and here Pusa sem2 and Pusa sem3 and Pusa 7. These are all some of the varieties produced, and they are able to resist which pest or which insect. Aphids, they are able to resist. Jacids, aphids, and other the fruit borers. Then shoot and fruit borers, they are able to prevent these varieties. So these varieties have been produced by our Indian agronomist, agriculturalist, or pomologist, you can call anyway, pomologist, those who are specializing on the fruit plants. And they are able to produce these by different crossing techniques. While you are crossing, you can take the cultivated varieties or the germplasm or the wild varieties. You can do by any method, and then you induce this disease resistant character into these plants so that they become automatically resistant to some of the insects. Plant breeding for improved food quality. More than 840 million people have no adequate food to meet their daily nutritional needs. See, India, I used to tell very often, India is a poor country, and then we are in the developing stage. We are also a developing country, and there are so many people who are going to the bed without any adequate food. I tell you, so many children in our country have never tasted the milk in their life, except the mother's milk. They have never tasted a cow's milk. And the babies, many children, they have never seen a hen's egg. Okay, and they have not eaten any chicken or mutton as the well-to-do people are doing. They are living in a below poverty line. We are not able to give an adequate food for them. And particularly the nutritional food is only a dream for them. Even a normal food they don't get. Then why to ask about the nutritional food? They are not having even a 3-time meal a day. They are not able to have even a 3-time meal a day. Around 3 billion people suffer from hidden hunger. Inadequate micronutrient, protein and vitamins because they can't afford to purchase enough fruits, fish and meat. Increased risk for diseases due to diets lacking in essential micronutrients, iron, vitamin A, iodine and zinc. It reduces their lifespan and mental disabilities. So these are all the problems that we are having in our country, particularly for our Indian babies and Indian children. They don't have an adequate food. So what we have to do, we have to improve the quality of the food by the plant breeding techniques. Biofortification is a beautiful topic once again. So what is this biofortification? Breeding of crops with higher levels of vitamins and minerals, protein and other healthier fats to improve health of the people. So that is what is called as a biofortification. A beautiful concept in biology. So you are breeding a plant, with the aim of increasing the vitamin level, mineral level, protein content and higher healthier fat. That you have to understand, healthier fat, not a simply fat, to improve the health of the people. Breeding for improved nutritional quality is to improve the protein quality of the and content, oil content and quality, vitamin content and then micronutrient and mineral content. So these are the things which we want to improve. We want to improve the protein, we want to improve the oil, we want to improve the vitamins, then micronutrients and minerals. So when you want to improve all these things, automatically it will become a healthier crop. So the production of the healthier crops in terms of vitamins, minerals, protein, fats, etc. is what is called the biofortification. Development of maize hybrids that had twice the quality of lysine and tryptophan compared to existing maize varieties. So these are all some of the examples. Development of improved cultivated wheat using Atlas 66 variety having high protein as a donor. Development of an iron fortified rice variety with over 5 times as much as a cotton variety and a golden rice with enriched beta carotene. These are all some of the examples. Atlas 66 variety, then golden rice variety, and in a maize the lysine and tryptophan amino acid increased to a great fold. Then some more benefits. Release of following vegetable crops by IARI, Indian Agricultural Research Institute, enriched with vitamins and minerals. Vitamin A enriched carrots, spinach and pumpkin. Vitamin C enriched in bitter gourd, batua, mustard and tomato. Iron and calcium enriched in beans lab means beans, then French beans and garden peas. Cultivation of mushrooms with high protein content in large scale in the agaricus. These are all some of the examples of the biofortification. Then we are coming to a next concept, what is called as the single cell protein, popularly called as the SCP, single cell protein. What's a single cell protein? It is an alternate source of a protein for animal and human nutrition in single cell protein because of the following reasons. It is the conventional agricultural production of cereals, pulses, fruits and vegetables do not meet the demand of ever increasing human and animal population. Shift from grain to meat diets, a very beautiful concept. Shift from a grain to meat. First we are depending on grain. Now when man began to think that if you are going to depend on the meat directly, then the consumption will be reduced. When you are eating the grains, you will eat a lot. But if you are going to eat a meat, the consumption will be only a very less. So man shifted his mode of intake from a grain intake to the meat intake. It results from the requirement of 3 to 10 kg of grain to produce 1 kg of meat by animal farming. See, if you want to produce 1 kg of meat, you have to spend 3 to 10 kg of grains. So now we are turning our attention to the single cell protein or SCP. More than 25% of human population is suffering from hunger and malnutrition. One of the microbes, cyanobacteria, spirulina and orthospira, can be grown on large scale easily on materials such as wastewater from potato processing plants, there is a starch plants, straw molasses and animal manure and even sewage. It serves as a food rich in protein, minerals, fat, carbohydrates and vitamins. It reduces environmental pollution also. Comparison. Now we will try to compare this, then you will understand how beneficial this biofertilizer is. 200 gram of a protein produced by a cow weighing 250 kg compared to the production of 25 tons of a protein by 250 grams of a microorganism. See, you need a cow which weighs 250 kg. The weight of the cow is 250 kg. You need that cow which will be producing 200 grams of a protein. So 200 grams of a protein is being produced by 250 kg the weight. Whereas now you see 25 tons of a protein is being produced. 25 tons means this much kilos. 1000 kilos make a ton. 25,000 kg of a protein produced by 250 grams of a microorganisms. Say this is methylophilus and methylotrophus because of its high rate of biomass production and growth. Crop disease and control. There is a heavy loss of crop plants and a considerable damage to the crops due to disease in the crop plants. It is necessary to control plant diseases by knowing cause of the diseases, life history and the causal organisms and meteorological conditions which influence a host and parasite interaction. The last slide in our show. Control measures. How to control the diseases in the plants. It's divided into two groups, prophylaxis and disease resistance. Prophylaxis is a protection of a host from the exposure to the pathogen, infection or from unfavorable environmental conditions. You see, you protect the plant itself and don't expose it to the disease and the pathogen. That's one method. The other one is disease resistance. Improvement of a disease resistance or a host to infection and to decrease the incidence of diseases in the plants. Okay, fine. Now, altogether I have given 3 lectures so far in the crop improvement or in the food production. Food production divided into 2 lectures, and now in this we were able to discuss something about the biofertilizers. Not only by the breeding techniques, but also by using the latest fertilizers, biofertilizers in the form of the microorganisms and also in different farms, we are able to improve the quality of the crops. How this is being done successfully by our scientists, by our farmers, is what I was telling you in the last 3 classes under the topic called food production. Thank you very much. Any doubt in this, any comments that you want to pass about this, you can share with me. You are always welcome. Thank you very much, my dear students. Thank you.